kinetics against SARS-CoV-2, which is the published, have published, is preprint on medRxiv. When I was postdoc in Dr. Cray's labs and the other ones, I'm going to just mention very fast my ongoing projects, because in pharmacology, which is preclinical pharmacokinetic model, investigating drug metabolism, and the other one is the genomics of tobacco use and concurrent psychiatric disorders. So two somehow very different from modeling. And it is, I'm just mentioning for the second one because we have used lots of statistical method. So I found it maybe appropriate. Oh, sorry. Okay. So this is our preprint insights into B cells and antibody kinetics using mathematical modeling. So our aim in this research is to develop a mathematical model to understand the dynamics and mechanism of humoral response to SARS-CoV-2. Specifically, we focus on the B cells and IgG neutralizing antibodies. We analyze the initiation of B cell response, including their differentiation to germinal center B cells, short-lived plasmablasts, long-lived plasma cells, and memory cells. And we are interested to identify the key parameters that are influencing the primary and the secondary immune response. So before starting, I would like to give a very fast introduction. As you know much better than me, this is we have two kinds of immune system, innate and adaptive. So innate immunity actually is the first part, is the body's first line of the defense against pathogens already present in the body. That is general, non-specific to the virus. It is rapid, limited, and low potency, and no memory. And it has some, it has some main components here, which is the natural killer cells, dendritic cells for the innate immunity. But we have also adaptive immunity, which is the opposite of the innate. It is built up, we are exposed to disease or get vaccinated. It is specific to the virus, for example, for HIV or SARS-CoV-2. It is slow in order to be activated. It's high potency and long-term memory it has. And the main parts of the adaptive immunity, we can say it is B cells and T cells. And also in the adaptive immunity, it's in two parts, which is humoral immunity and also cell-cell interaction immunity. So in this paper, we actually focus on the humoral immunity, which is the antibody-mediated immunity. And we studied the B cell activation in primary response and the secondary response. So as you can see in this schematic figure created by render, through phase 1 and phase 2, first of all, naive B cells are activated, encountering with pathogen. And then they have some T cell interaction here, and then activated again and differentiated to the short-lived plasma B cells and the germinal center B cells that are main focus in this paper. Then germinal center B cells, through phase 2, they go to the dark zone and light zones of the germinal center. In dark zone, they are doing the somatic hypermutation. And in light zone, again, they are super activated by encountering with virus and also interaction with T follicular helper cells through the interleukin-4 cytokine. So we focus on interleukin-4 because we have these parameter values from that, and we ignore some other interleukins that are important, like interleukin-21, but we didn't have parameter values for that. So anyway, the germinal center B cells are differentiated to the memory B cells and long-lived plasma B cells that are important cells. And in the secondary response, the secondary exposure to the virus, the memory B cells now active, they will get activated and differentiated to the long-lived plasma B cells and the germinal center B cells. This is the whole biology behind. So based on all these details, we developed our ODE equations, actually delay differential DDE equations. So our equations have 3 parts. First part is the virus host pathogen actually interaction, which have some DD equations. And then for the second part was primary immune response and secondary immune response. I didn't include any equations here just I'm focusing on the figures. So in these equations, as you can see, this figure here, the small one here, is showing the symmetric and asymmetric division of the cells. So the germinal center B cells, as you can see here, they can differentiate, they have self-renewal probability PB, and then 1 minus PP, they are differentiating. And as well, we can see by probability PP, they are differentiating to the plasma B cells, 1 minus PP memory B cells. And as well, for the memory B cells, we have some probability that they are doing symmetric and asymmetric divisions. That is from the one of Dr. Komarov's papers. So this was biology and the equations. So now the main part in our research was to focus on the neutralization effects of the antibodies through the Michaelis-Menten kinetics, this equation here. So we have one OD equation for the viral load, and we added this term in order to show the neutralization effect of the antibody. So in this kinetics, we have 3 main parameters, which is the Emax, IC50, and H. So Emax is the maximum saturated level of the neutralization. Here it is neutralization, and that is the maximum level of the neutralization. And IC50 is the number of antibody that we reach to the 50% of this neutralization. And parameter H is the Hilo coefficient, which is also cooperative of the antibodies. So we fitted this equation only in our equations with the clinical data with Wuhan and Omicron, 2 different variants of the SARS-CoV-2, and we found different values. So for Emax, for Wuhan, you can see the values there is a little bit higher than Omicron. But the IC50, if you look at in this equation, so the IC50 for Omicron is higher than the Wuhan. So it means that we need more antibody for Omicron infection than Wuhan. It means we need more neutralization for Omicron. So in the other side, we can say, so, okay, Omicron is more immunoevasive because we need more antibody to neutralize it and to kill the viruses. And the Hilo coefficients or cooperative effect is higher for viral load, which is 1.2 in comparison with Omicron. And again, these are all showing that Omicron is more immunoevasive. So these are our parameters that fitted, but as I mentioned, these are only parameters in our big models that we fitted, 3 parameters based on the data. We have another parameters that are all coming from the literatures, reliable literatures, and some assumptions that we have done for the parameters. What is the assumptions? In our equations, we have assumed that in the primary response, antibody are generated by short-lived plasma B cells and long-lived plasma B cells. And because our equations are very simple, so we have only assumed the proliferation rate of antibody by short-lived plasma B cells and long-lived plasma B cells. And in secondary immune response, we have long-lived plasma B cells only. Therefore, we assumed some assumptions for this antibody and then compared our model prediction with 32 clinical data. And if you look at this paper, so it shows that our model is in good agreement with the data. And as well, we compared with the viral. In the viral load, we had only 6 data points. And then we compared with Wuhan and Omicron. And we can see that for Omicron, viral load is a little bit higher than the Wuhan. So after this, we did sensitivity analysis, global sensitivity analysis. We assumed 1,000 hypercube sampling for each parameter, changing all together. And then we looked at the antibody peak values and antibody peak time, which is our main focus in this paper. So yeah, I listed some sensitivity of this, but I'm not going to go through them, but all makes sense if you go through that. But the main one is that antibody, the peak antibody level is sensitive, responsive to the antibody production rate of the short-lived plasmablast and long-lived plasma B cells in the primary response. And you can, this is based on Spearman correlation coefficients. We can see the alpha P, which is the generation rate of the antibody by long-lived and short-lived plasma B cells. It is about, for long-lived even, it is a little bit higher than the short-lived plasma B cells. So then we also did the sensitivity analysis for the secondary immune response. And if you look at here in the bar chart, so the peak antibody is very sensitive for the generation rate of antibody by long-lived plasma B cells. It has 77% Spearman correlation coefficients. So, okay, so we find these parameters that are important in finding the antibody level. So then, in order to find the minimum, maximum, to capture minimum and maximum, and also dashed line is for secondary, we focus on those parameters, antibody generation rate. And we saw that in order to achieve the secondary immune response the same as the primary immune response, antibody response, we need to have 10-fold higher antibody generation rate by plasma B cells in secondary in comparison with the primary. This number is completely random because reviewer also asked me to expand, to give some explanation about that parameter 10-fold, but it was just random. I just increased the secondary antibody generation rate in order to achieve the same antibody level. But the point is that our model only have the antibody generation rate. We didn't include any affinity coefficients in our equations. So this is the limitation of our model because we don't have any affinity coefficients. We should have this because we know that from biology in the secondary immune response, the antibody response or the secondary immune response is stronger than the primary immune response. So the coefficients of the antibody should be for sure different. But since we don't have, we just say that in the secondary immune response, we have more antibody generation. And then as a last result, this is, we looked at the neutralization effect, neutralization for the Wuhan and Omicron in the primary response. We can see that for Wuhan, it is higher than the Omicron. And also we look at the different times, different scenarios for the secondary response, which is Wuhan, 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 Omicron, Omicron, Omicron. We can see for the Wuhan, Wuhan scenario in the secondary response, the neutralization is always higher than the Wuhan, Omicron, or Omicron, Omicron. And also we looked at the different time period, like 3 months, 6 months, 1 year later than the primary response for the secondary exposure. So we can see the later time that we have, the lower neutralization. For 1 year secondary exposure, it has the lower neutralization. And as well, lower antibody level. We can see this green curve is higher because that is 3 months later. So it shows waning immunity. During the time, we have waning immunity. And if you look at the viral load for two different scenarios of Wuhan, Wuhan, and Wuhan Omicron, we can see for the Wuhan Omicron scenario, Wuhan is primary, secondary, Wuhan Omicron. So the viral load for the Omicron is higher than the Wuhan. Again, this is immunoevasive of the virus. Omicron. So this was for the first time. And I'm going very fast for this ongoing project. So preclinical pharmacokinetic model. This is a project that I have started. It's about 2 months that I'm working on that. In this equation, in this project, we have data for the African green monkey, which is a preclinical model. We have age and we are interested to look at the age and sex differences on drug metabolism. So the drugs that we are looking currently is nicotine, coumarin, and dextromethorphan, and these are parent substrate, and also we have some metabolite that first we are focusing on the parents. So we are looking at this the concentration of this drug during the time, and then for different ages, different age groups, and female and male. And then so far we have identified 7 pharmacokinetic parameters that we have, because when we say the pharmacokinetic, it means that absorption, distribution, metabolism, and excretion. So for these 4 scenarios in pharmacokinetic, we need 7 parameters, which is Cmax, is the higher, you can see in this figure, it is the higher concentration of the drug, Cmax, and the Tmax, and the half-life of the drug. And also the AUC, area under the curve, which is the total exposure of the drug, and the clearance rate, and yes, 7 parameters. So based on data, we have these parameters, but our interest is to develop population PK model, because we have intra and inter diversity in our population. So we would like to capture all this in our model, and then we need to work on the population PK model that I haven't developed yet. So you can see this diversity in our, I think it's, I'm running out of time. Okay, I'm going to just, yes, these are the diversity for female and male for the just nicotine concentration for different age. You can see the diversity because these are the standard deviation that are included in these bars. So in the second part, I'm just mentioning here that many features are shared between substance use and psychiatric disorders. So our interest is to find the genetic liability between schizophrenia and smoking behaviors, because they have smoking behaviors and schizophrenia. Some have symptoms, same symptoms. And also the main one is that the heritability of smoking is 50%, twin-based heritability, and for schizophrenia is 80%. And we would like to see that which one causes the other one. And based on the genetic liabilities, and which is very important. And you can see that even for the smoking, daily tobacco users have twofold higher risk of developing schizophrenia. And then after that, we would like to think about the drug development and anything. So these are all statistical methods that I have used. And then in the last part, you can see, and with that, the main statistical method here is that I have used multi-rate analysis. It means that instead of looking at schizophrenia separately, and instead of looking at, for example, cigarette per day or smoking initiation, smoking cessation separately, we combine all these traits together, and then this leverages the genetic liability and SNPs that they have together. And we can see that it is the risk factor has been increased from 1 to 7. It's very interesting. It says that when we are looking at this cigarette per day based on the schizophrenia data. So I'm going to thank my supervisor, Dr. Tyndall, Dr. Chenoweth, and also Dr. Morgan Craig, that I was postdoc in her lab, and also the organizer of this conference. Thank you.
Okay, thank you, Suzan.